In this video, we'll review basic information on intermolecular forces, also referred to as intermolecular interactions or attractions. What is important to know is that intermolecular forces are non-covalent attractions between atoms and molecules. And the key to this is that they are non-covalent, meaning that the atoms will not share electrons that will interact with the nuclei of both atoms. These intermolecular forces are important in key processes of life, such as folding of proteins, recognition of substrates by enzymes, DNA replication, and many more biochemical processes. We'll review the basic characteristics about three very important forms of intermolecular forces called ionic bonds, also known as salt bridges, hydrophobic interactions, and hydrogen bonding. Hydrophobic interactions are known to be the major driving force in protein folding, while hydrogen bonds and salt bridges are important in protein folding, function, stability, and flexibility of the protein. In this video, we will talk about these three intermolecular forces. To talk about these forms of interactions, we are going to be using the protein myoglobin as an example to show these interactions taking place in an actual protein. Myoglobin is the protein in charge of carrying oxygen to the muscles. Here you can see the three-dimensional crystal structure of myoglobin. The main chain of myoglobin is folded into 8 alpha helices, which is about 70% of the structure, and the other 30% is mainly formed of loops and turns. One striking fact about the interior of this protein is that, for the most part, it consists almost entirely of nonpolar residues, such as alanine, valine, leucine, isoleucine, and phenylalanine. For the most part, charged residues such as aspartate, glutamate, lysine, and arginine are on the outside mixed with some other polar residues, such as serine, tyrosine, and glutamine, these along with other nonpolar residues. The only polar residue inside of the myoglobin is histidine, which together with heme allows the binding of oxygen to be delivered to the muscles. Later on, we'll zoom into different parts of this protein to see what type of intermolecular forces are contributing to the three-dimensional structure of this protein. First, let's talk about hydrophobic interactions. Like I mentioned at the beginning of this video, hydrophobic interactions are known to be a major driving force in protein folding. These interactions occur among nonpolar substances to minimize their contact with water and/or amphipathic molecule. This image depicts how hydrophilic polar residues are on the surface of the protein, while the hydrophobic, nonpolar residues are buried in the inside of the protein. In the example with myoglobin, we can see 4 nonpolar amino acid residues buried inside the protein, surrounded by alpha helices. To be more specific, as you can see with the Lewis structures and three-dimensional structure, there are a series of residues that are interacting with each other, avoiding the interaction with water molecules. There are 2 side chains of isoleucine, isoleucine 101, and isoleucine 142. There is 1 leucine amino acid residues, leucine 104, and 1 phenylalanine, phenylalanine 138. As you can see, the main chains of these amino acid residues are more exposed to the surface of the protein, where they can interact with water molecules, since the main chain of the protein is polar, containing an amine and carbonyl group. The next interaction that we'll talk about is hydrogen bonds. Hydrogen bonds are strong attractions between the hydrogen atom and a neighboring molecule containing a fluorine, oxygen, or nitrogen atom. The large electronegative difference between hydrogen and any of these small electronegative atoms causes the hydrogen atom to have a fairly large partial positive charge within the bond, while the fluorine, oxygen, or nitrogen atoms have a fairly large partially negative charge. In the representation below, you can see an example of hydrogen bonding, where one water molecule is forming hydrogen bonds with two molecules of water. Hydrogen bonds are usually represented by dashed lines like you can see here. One very important aspect about hydrogen bonds is that they should not be confused with chemical bonds. Chemical bonds occur between individual atoms within a molecule, whereas hydrogen bonds are intermolecular forces that occur between molecules. One example of hydrogen bonding in myoglobin is the interaction between serine 3,5 and arginine 3,1. The oxygen from the hydroxyl group interacts with a hydrogen from the guanidino group of arginine 3,1. In this two-dimensional structure, the interaction is represented by the dashed line between oxygen and hydrogen. In the three-dimensional structure, you can see that the hydroxyl group of serine 3,5 is 2.7 angstroms from the hydrogen atom of the guanidino group from arginine 3,1. Keep in mind that interactions between residues only occur for distances shorter than 3.5 angstroms. Using the measurement tool in PyMOL is a good way to make predictions about possible hydrogen bonds within the proteins. The association of two ionic charged amino acid groups of opposite charges is known as a salt bridge, or ionic interaction. For this interaction to take place, the side chain of the charged group of atoms has to be at least 3.5 angstroms from each other, just like the hydrogen bonds, and at least one side chain of aspartate or glutamate side chain of carboxyl oxygen, and a side chain of nitrogen atom from lysine, arginine, or histidine needs to be present. These amino acids are charged at pH 7 due to their side chain pKa ranging from 3.90 to 4.10 for the negatively charged residues, and the positively charged residues pKa ranging from 6.0 to 12.5. In the Lewis structures, we can see that lysine 7,7 is protonated at pH 7 since its pKa is about 10.5, making it positively charged, whereas glutamate is negatively charged. This is the deprotonated form of glutamic acid with a pKa of about 4.1. The presence of these two oppositely charged groups allow for an ionic interaction to take place if they are at least 3.5 angstroms from each other. In the three-dimensional structure of the proteins, lysine and glutamate were highlighted as stick figures. The blue end of lysine represents the presence of a nitrogen atom at that end. As for the glutamate, the two red ends represent the presence of oxygen atoms. Because at pH 7, glutamate is deprotonated, both bonds to oxygen in the glutamate are identical, indicating that the electrons are shared between the two bonds. For this reason, in the Lewis structure, a resonance hybrid of glutamate is shown to represent the electron delocalization in the structure, and the formal charge in the middle. Unlike hydrogen bonds, no dashed lines are used to represent the interaction, just the presence of positive or negative charge symbols. In the three-dimensional structure, we can see that the oppositely charged side chain groups are approximately 3.1 angstroms from each other, allowing a salt bridge to take place. As you can see, There are three main intermolecular forces in proteins: ionic interactions, also known as salt bridges, hydrogen bonds, and hydrophobic interactions. These serve a crucial role in key processes of life, such as folding of proteins, recognition of substrates by enzymes, DNA replication, and interaction of proteins.